Hello everyone, my name is MoXia and I am doing my master course in Saitama Institute of Technology in Japan. Today I am going to give a presentation of my paper, CNN Recognition of Epileptic Foci based on Composite Signal Processing of EEG Data. Here is the outline of my presentation. I will introduce some background related to our research. Then I will show the objective and the methods of our works. After that is the experiments and results, and finally the conclusions. Here comes the background. Epilepsy is a chronic disease caused by sudden abnormal discharges of neurons in the brain, which lead to transient brain dysfunction. Epilepsy are generally fall into two main types, focal seizure and generalized seizure. Epilepsy is one of the most common neurological diseases and occurs between 40 to 70 per 100,000 people in the general population. About 70% of patients can be treated with medication. However, for patients with drug-resistant focal epilepsy, the only way to prevent epileptic seizures is to surgically remove epileptogenic tissues in brain, which means localization of epileptic focus becomes an important task in surgical treatment. So far, monitoring of patients' EEG is the main basis for determining to localize the seizure areas of epilepsy. EEG is the electrical activity collected by electrodes placed on the scalp and represents the macroscopic activity of the surface layer of the brain underneath. During seizures, the signals produced by abnormal discharge in epileptogenic zones on brains are called focal signals, and the signals produced by normal discharge in non-epileptogenic zones are called non-focal signals. Because the seizures are caused by uncontrolled abnormal discharges in the focal areas of the brain, the EEG signal of the focal and non-focal areas will be different during seizures. Medical professionals can localize the focal area by comparing the EEG of different areas of the brain. Traditional diagnostic method is visual judgment on EEG by experienced doctors. However, this method has two main disadvantages. The first disadvantage is that the implementation of this method is extremely dependent on trained and experienced doctors, which are not available in every hospital. Another disadvantage is that the volume of data of long multi-channel EEG record is very large and that visual recognition process requires big amount of time and effort of the doctors. Therefore, an automatic recognition of focal localization as a support technology for doctors is required. The key challenging problem lies in the accurate classification of focal and non-focal EEG signal. What we did in this paper is to achieve classification of EEG signal recorded from epilepsy patients by applying deep learning. This figure shows the process of EEG classification using machine learning. Previous experiments have demonstrated that using data processed by feature extraction for training increases the accuracy of machine learning models rather than inputting raw data directly into machine learning training. In our experiment, we choose convolutional neural network, a typical machine learning technology, for the classification of focal and non-focal EEG. The reason why we choose it will be explained later. There are many signal processing methods used to perform feature extraction, but usually only one of them is selected in past experiments. Our experiment have modified the original process of classification. In the signal processing stage, we used two different feature extraction methods on the same signal, and two different feature maps can be obtained due to different feature extraction principles. After that, we combine the two-dimensional feature maps and use the combination as training data. This way, the training data contains more features for the model to capture, and we hope to improve the accuracy of the training results of convolutional neural network. Next, I will introduce the two feature extraction methods used in our experiment. The first method we use is the short-term Fourier transform. The main idea of STFT is regarding non-stationary signals as the superposition of a series of short-term stationary signals, and the short-term can be realized by adding certain windows along the time axis. Selecting a time frequency localized window function and assume that the analysis window function is stationary in a short time interval. Moving the window function so that every time there is a stationary signal in a finite time ways, and therefore to calculate the power spectrum in different times. Another feature extraction method is the continuous wavelet transform. Like Fourier transform, CWT is also a kind of integral transforms. Because the wavelet base has two parameters in terms of scale and displacement, expanding on the wavelet base means projecting a function versus time onto a two-dimensional time frequency phased plane. Also, because of the characteristics of the wavelet basis itself, after the function is projected into the wavelet transform domain, it is helpful to extract some features. Both methods we used transform one-dimensional signals to two-dimensional matrices. After using the two feature extraction methods, we need to combine the two types of feature maps. However, the result after CWT is 10 times bigger than STFT in terms of data size. Therefore, an individual pooling layer is used to compress feature map from CWT. Pooling layers are usually placed between continuous convolution layers in CNN structures to compress the amount of data and parameter to reduce overfitting, meanwhile reducing the matrix size by extracting key features out from the sources. The pooling method used here is max pooling because this step focuses on intense features in data rather than the global status. After combination, we can obtain a 2 times 1024 times 100 feature map for each one-dimensional signal. This is the main structure of our convolutional neural network. Also, I will explain the reason why we choose CNN for training here. Inspired by the high accuracy of VGG16 network in image recognition, we modified its structure and parameters to fit our requirements. VGG16 used red, blue, and green colors in each pixel as 3 channels to extract feature from 244 times 244 images through 16 convolution layers. In this experiment, the number of the channels have changed to 2, one channel for the STFT matrix and the other one for the CWT matrix. Meanwhile, the number of convolution layer and the corresponding kernel size were also adjusted to fit the new input size, 1024 times 100. The main CNN structure is formed by 4 convolution blocks, and each of the blocks contains 2 convolution layers, followed by activation functions and 1 max pooling layer. The kernel size of each layer and the data size between each block are shown in this figure. After the 4 convolution blocks, there are 5 fully connected layers, also followed by activation functions except the last one. The layer followed by the last fully connected layer is the dropout layer, a layer that randomly abandons 50% of nodes in fully connected layers to avoid overfitting. The function of this part is mapping all connected features into one dimension and complete the classification. The EEG signal data used in this paper is obtained from publicly available EEG dataset called Bern Barcelona EEG database. The data was collected from 5 epilepsy patients who underwent long-term intracranial EEG recordings. All patients were suffered from long-standing drug-resistant temporal lobe epilepsy and candidates for surgery. Signals recorded at epileptogenic zones and non-epileptogenic zones and were labeled as focal and non-focal respectively. Each class consists of 7,500 signals collected from adjacent channels in the same zone. All signals were collected during epilepsy seizures, and each signal has a sample frequency of 512 Hz and lasts 20 seconds. After completing the training, we tested the accuracy, and here comes the results. The machine learning model obtains the highest accuracy of 91.3% in validation. We also tested the data performed by STFT or CWT only with the exact same CNN structure except for the number of channels. And the accuracy was 87.7% and 86.2% respectively, both lower than the combined feature map. We have also compared our results with those of other experiments. Our accuracy exceeds some existing experiments based on EMD or entropy. Comparing with some other results which obtained by using multiple machine learning systems, our accuracy is lower. However, our methodology and training process requires less time and computing power to complete. The accuracy of the model trained by the combined data was higher than either model trained by signals merely performed by STFT or CWT. This result demonstrated that the combination of different feature maps from the same signal involved in machine learning can convincingly improve the accuracy of the model. In addition, the accuracy may have further increasing by adjusting and testing more parameters. Also, it is worthy trying to combine even more channels that come from other transforms or feature extraction methods for training in further works. That's all. Thank you for your attention.